Good morning, good afternoon for some folks, good evening for others. Welcome to a research update on gene therapy. We're thrilled to have attendees from around the world joining us on today's webinar. I'm Michelle Davis, Executive Director of the International FOP Association. Before we get started, we'll do a few Zoom housekeeping notes. This webinar offers translation services for those who are more comfortable listening in their native language. You'll click the down arrow if you'd like to participate in translation services. You'll click the down arrow at the top of your screen, click View Stream on Custom Live Streaming Service, select your language, and click Join. If you have technical issues during the call, please put that in the chat for the hosts. You are all muted so that background noise at each location doesn't disturb the webinar, but we do want your questions, so please ask your questions in the Q&A. They'll be answered at the end of Danielle's presentation. And then finally, the webinar is being recorded, so you can come back and listen again, and we'll be sharing this webinar on our website and social media channels so others can listen too. We hope you'll share it as well. We were excited to launch In Pursuit of a Cure in 2020, and we're thrilled to bring it back for a second year. Why? One word, progress. Actually, two words, exciting progress. A lot of progress has been made, and the learning support moving forward with the next phase of research. I'm also excited to introduce you to Dr. Danielle Kirkovich, who joined the IFOPA on April 1st of this year as Director of Research Development and Partnerships. Danielle joins the IFOPA after 10 years of serving as Principal Scientist for Beyond Batten Disease Foundation. Danielle received her doctoral and master's degrees in biomedical sciences from the Albert Einstein College of Medicine. She has extensive experience with government and foundation programs and has been involved with planning and projects that include post-war rehabilitation following polytrauma, spinal cord injury, and amputation, including experience with trauma-induced heterotopic ossification. Her experience in rare diseases and rare conditions ranges from fourth-degree burn injury to pediatric cancers and lysosomal diseases, including juvenile Batten disease, where she is responsible for bringing the first interventional treatment for the disease to a phase 2/3 trial planned for 2021. We are thrilled to have her expertise as both a scientist and a research director for another rare disease patient advocacy organization. Welcome, Danielle. We hear a lot about gene therapies in the news and on social media and how they are being used in diseases like FOP that are caused by a genetic mutation. Your master's degree included a focus on genetics. Could you tell us a little bit about the history of gene therapy?
Thank you, Michelle. I'm thrilled to be part of the IFOPA, and you just hit on one of my favorite topics other than scientific research, and that's history. Here is a photo of Friedrich Miescher in the late 1800s when scientists were still debating whether or not cells actually existed. In 1860, in his lab, shown on the right, Miescher discovered DNA, the main topic of our discussion today. In these pictures, we have Oswald Avery, Colin MacLeod, and Macklin McCarty, and they took things a steep step further, but not for a while, not until 1944. These men worked together to show that DNA specifically was responsible for inheritance. And you might recognize the DNA helix structure in bright colors on either side of this picture of James Watson and Francis Crick, who discovered the structure of DNA in 1953. But if you don't recognize the DNA, you might recognize this guy. In 1939, Bill Finger, before Avery, MacLeod, McCarty, Watson, and Crick, he introduced the concept of gene therapy to us all when Batman was created. But it wasn't until years later we learned his superpower gene was activated by a virus. And I'll tell you more on this later. Although Batman was conceptualized in 1939, it was Paul Berg in 1972 who made the Batman story plausible when he created the first artificially assembled DNA discovered by Miser and understood by Avery, McLeod, and McCarty, and pictured by Watson and Crick, when he packaged that DNA into a modified virus, which infected human bone marrow cells in a dish. So his work showed that artificially created DNA could be made, packaged, and delivered into human cells.
So Danielle, you said that Paul Berg packaged DNA into a modified virus. Why did he use a virus?
That's a good question. At this point, researchers had figured out the recipe for making DNA. What they didn't know was how to deliver it into the human body, where it could do some good. Because viruses are very good at infecting the human body, Paul Berg took a virus, cut out the instructions that make us sick, leaving room for some cargo, and that cargo was the improved DNA cargo, and used the modified virus to deliver the new DNA. And there we have gene therapy.
Wow. So when did gene therapy trials start in humans, and did they use modified viruses?
Yes, the first gene therapy was actually in 1990, and the first clinical trial was conducted using modified viruses, and the patient was a 4-year-old girl named Ashanti De Silva, who had severe combined immunodeficiency, or SCID. In Ashanti's case, the disease is caused by the absence of a gene, which made a protein necessary to fight infection. Here she is at the NIH, the National Institutes of Health in the United States, with her brother and sister before gene therapy. She could not be exposed to anything. And here she is in 2018 in her profile picture. She is a writer in Chicago, Illinois, in the US today. Oh. And so how did they do this? First, they took stem cells, removed them from her body. The correct DNA was added in the laboratory, and a modified virus was used to carry that DNA, the DNA she was missing, the good DNA, back into her body, and she is fine today. Over $50 billion in spending and 3,600 registered clinical trials and 120,000 scientific publications yet later, there are now 22 approved gene therapies worldwide.
Are the approved gene therapies all the same, or are there different types of gene therapy?
The original definition of gene therapy is addition, shown here as number 1. But now that definition has been expanded. The definition now includes gene silencing, where one turns off problem genes caused by a defective gene, or gene replacement, which is a combination of the two, where we add good genes while silencing defective genes that are causing a problem. There is a 4th category, gene editing, that we'll also talk about.
So is gene therapy the right choice for FOP, and which type or maybe types will work?
The short answer is we don't know.
So how do we find out?
First, let's take a look at why FOP happens. It's a lot different than replacing a missing gene, as in Ashanti's case. Since for most everyone, it's been a long time since we've had a science class, I'll start with a little bit of a backstory.
It's been since 1993 for me, so refresh me.
No problem. Cells are the building blocks of our body. Think of DNA as a biological blueprint or set of instructions telling the body how and when to make different cells, where to put them, and instructing those cells how to interact with one another over time and different conditions. Some like to refer to cells as biological building blocks, much like Legos gathered here to make something remarkable. But how do cells position themselves in the body, decide what kind of cell to become, and then carry out their functions? Unlike Legos, each step isn't the work of a master craftsman, and the work isn't done when the body is created. Even when we sleep, our 30 billion cells are hard at work, reacting to one another in their own microenvironments, fine-tuning their actions. Imagine this picture is one of those Lego-like cells, a stem cell, if you will. A stem cell is the large light blue picture here, and it's waiting for instructions to tell it where to go and what to do. Pictured in a darker blue is the receptor, or a receptor protein. Receptors are proteins that sit partially inside the cell, partially outside the cell. The cell receives its instructions when substances, often called ligands, outside the cell bind to the outside of the receptor and transfer a message from the outside of the cell to the inside of the cell, starting a chain reaction to tell that cell what to do. In the instance of a stem cell, what kind of cell to become. So now let's look at that same picture, but now let's name a few things. So now the receptor is the ALK2 receptor, which is healthy and normal in most of us, but is not healthy in FOP. The ligand that the ALK2 receptor, or the substance outside the cell it responds to, is bone morphogenic protein, most often called BMP, and a substance that is hanging around whenever there's trauma, but a healthy ALK2 receptor does not respond to, is activin A. So when we take a look at this next picture, here we have the ALK2 receptor in black, and I put it in black to show that it's dysfunctional. There's something wrong with it. The ACVR1 DNA had a mistake in it, and it made that mistake when it made the receptor. Now what we have is a receptor that's hypersensitive to the BMP ligand, but it's newly sensitive to activin A. And because activin A floods an area of cells whenever there is trauma, the activin A binds the receptor, and now the message that was a little bit quiet before that said make bone is now enormous. And the message is make bone, make bone, make bone. And at the same time, there are messages telling those cells to divide and telling all of those cells to make bone. So the question was asked, can researchers use gene therapy to replace the dysfunctional ACVR1 gene that makes the hypersensitive mutant receptors so that activin A does not have the power to turn stem cells into bone-making cells?
So as many on this webinar are well aware, because their donations and fundraisers have made it possible, the IFOPA has been funding FOP research since 1990 with a special focus on drug development for FOP since 2015. That's our Accelerating Cures and Treatments, or ACT for FOP grant program. The potential drug therapies being explored in that program fix pieces and parts of FOP, but don't correct the root of the problem, the ACVR1 gene. So in 2020, the IFOPA asked whether researchers could use gene therapy to replace dysfunctional ACVR1 genes that make mutant receptors, those black receptors you just saw on Danielle's slides. We turned to gene therapy experts at the University of Massachusetts to help us answer that question. Together, the ACT for FOP Drug Development Grant Program and our Gene Therapy and FOP Research Program comprise our In Pursuit of a Cure campaign. To help advise our gene therapy and FOP research program, the IFOPA developed a gene therapy working group to bring the best and the brightest in FOP, bone and cell biology, and gene therapy together. You probably recognize a few faces in this group. And the FOP community, many of you who are on this webinar today, answered the call to action to financially support the work of the investigators at UMass. So Danielle, tell us how the UMass researchers got started.
So the researchers at UMass got started by looking at a series of strategies, and they devised a plan using 3 different strategies in gene therapy that might work for FOP. FOP is a dominant gain of function mutation. That means that the dysfunctional ALK2 receptor proteins are not simply quiet and non-functional or non-existent, as in Ashanti's case, where her missing gene was added. Mutant ALK2 receptors are present and they are causing a problem. So the investigators asked themselves, could they solve the problem by adding healthy ACVR1 genes? Could they solve the problem by silencing mutant ACVR1 genes? And what if they did both? So to recap, FOP stem cells have both individuals with FOP actually have healthy ALK2 receptors. That's something we don't talk about a lot. But what we have is, because you have two genes and one gene is mutant or has a mistake, or then half of your genes that are supposed to make ALK2 receptors make healthy ALK2 receptors, and the other half make the mutant receptors. So each cell, even though in the earlier pictures we only showed one receptor in one cell, each of our cells carries 10 to 20,000 receptors. So there's a crowd of action going on here. So to recap, in the left, FOP stem cells have both blue, healthy, and black mutant ALK2 receptor proteins on the left. I'm sorry, I think that we should be looking at gene addition. Sorry.
Oh, I think we missed the feasibility study slide, Danielle. I don't know if you can go back one slide where we talked about the things that they were going to focus on. So maybe we can jump back and do that before we jump into how it went. No problem.
Okay. Yeah. I got ahead of myself because I'm excited about this. Okay. So in the first year, let's just pretend we didn't see that. But in the first year, we have feasibility studies that researchers at the University of Massachusetts did. One, they wanted to know which viral vector is best for carrying the ACVR1 good gene. The ACVR1 mutant silencing sequence, or both. They simply needed to know which carrier was best. Second, they needed to look at silencing sequence. We already know the best DNA sequence for the ACVR1 receptor. It's the healthy one that most of us have. So, but we didn't know the silencing sequence. And so researchers looked at several silencing sequences to determine which blocks the function of the mutant ACVR1 gene and ultimately the mutant receptor, but not the healthy one. And third, once they had determined that, they needed to know which vector, gene, and silencing combinations that they had explored in 1 and 2 actually entered into FOP patient cells in a dish. So this is where the rubber is really hitting the road. We can make a wonderful project on paper, but it's got to work. And fourth, this was the big part. They wanted to know which vector silencing sequence or combination entered FOP mouse cells and worked in vivo in a live animal. There's an awful lot going on, and we needed to make sure that it got there and it did what it was supposed to do. So now I'll jump ahead. See, I was excited. So on the left, we've got FOP cells that are half mutant receptors and half healthy receptors. And gene addition, what they wanted to do is simply add more good receptors or add more good gene. And if we could flood the cells with good genes and therefore good receptors shown in blue, would it sort of overcrowd the action that the mutant receptors wanted to take? Next slide, please. So the next was, okay, one was to overcrowd with good genes. How about we just silence the bad genes? Would everything be okay? Would we shift to health if we simply silenced the bad genes? Next slide, please. And what if we did both? What if we added more good genes, and at the same time added silencing sequences, what would give us the best response in the animal and in human cells in a dish? So let's take a look.
Okay, so we are going to jump up for 1 second to a different slide.
Yeah, we had a little technical difficulty showing you the slide, but we thought this was so important that we would have a glitch in the system while we pulled that up. So if you take a look on the left of your screen, you'll see 3 mice. You'll see 1 mouse in the lower right corner, and it's not moving very much. That's an FOP mouse model. It definitely has FOP, it has extra bone, it's not moving very much, and its jaw is locked. If you look at the other 2 mice, the one that is sort of sticking to the right-hand column, we had arrows, but they went away, but sticking to the right-hand side of the cage, that animal never had FOP. It was always healthy, so it's running around. The third one that's staying sort of in the middle, that's an FOP mouse that has been treated with gene therapy in the combination. So the combination of addition and silencing worked best. It got into human cells in a dish, and it works in the mouse model.
Wow, that is exciting. But it is important to remember that there is a big jump from seeing something work in mice to knowing that it is safe and works in humans. But these results tell us that to stay in pursuit, to keep researching. The funding the FOP community provided accomplished a lot of positive results in year 1. So what's the next step? What questions will be answered in year 2?
Thank you. These are really important. It is exciting to see the mice, but there's a long road ahead of us. So encouraged by these promising results, there's an ambitious plan for year 2, which is why we need to raise additional funds. What the researchers answered in year 1 is whether or not it could be done. The answer is yes. It can be done in FOP patient cells in a dish in the laboratory and FOP mouse models. Year 1 was all about feasibility. Year 2 is all about fine-tuning the program for safety and efficacy in humans. So first of all, researchers need to ask themselves what the effects of gene therapy could have on a human immune system. If a person has an immune reaction to the gene therapy, this could be really bad. If they have one, at the very least, it knocks out the gene therapy and you haven't done anything. At the very worst, someone could get very sick. So it's really important that we take a look at that. The second part is we were only looking to see if the animal moved and what was happening in the skeleton. But what we really need to do is investigate what's happening in the rest of the body. We don't want to cure the skeleton and hurt the heart. And third, researchers needed to know which promoter to use. So a promoter is a really interesting piece of DNA that usually sits right in front of a gene, right in front of the ACVR1 gene. And it's like a dimmer switch. It tells the ACVR1 gene how much ALK2 receptor protein to make. Make a little, make a lot. And sometimes the promoters that we use in therapy are a little bit higher or lower or stronger or dimmer than those that we use in gene therapy. In normal situations, gene therapy, sometimes we need to use a different one in order to get the best health outcome. And lastly, what I wanted to mention, what I mentioned just touched upon earlier, is that the team at University of Massachusetts will also be exploring gene editing, which is very different than addition, silencing, or the combination. FOP is most often the result of a single change in DNA sequence. In the classic FOP mutation, there is a guanine base where there is supposed to be an adenine base. And this is what results in the mutant ALK2 receptor. Researchers want to determine through gene editing whether the newly discovered base editors capable of precisely changing one DNA base to another can be used to reverse the single DNA change that causes FOP and return it back to normal. Thus far, there are 7 gene therapies that have been approved worldwide using this kind of editing technique. However, all of the editing takes place in a dish. So cells are taken out of the human body, fixed in a dish, and put back in. For manipulations outside of the body, the number of approved therapies is growing. But for FOP, where we want to fix cells in the body, throughout the body, CRISPR-Cas9, which you may have heard of, base editing techniques are unpredictable. We don't know where that editing tool is going to travel in your body and what it's going to do. So we really need to take a step back and ask ourselves, how do we make the leap from being able to do it in a dish and do it in your body? But we have some exciting this is a recent paper in the last 2 months. Researchers are starting to harness CRISPR technology. Shown here is a mouse model of genetic deafness caused by a point mutation, much like FOP, where investigators injected a base editor delivered by the same virus the University of Massachusetts researchers are using in their studies. And the treatment reversed the mutation and partially restored hearing in the mice. So this was not corrected outside the body. The base editor, the technology, was directly injected into the animal, and the animal can now hear.
Wow, there are incredible advances being made in gene therapy. It's exciting to see those learnings and experiences being applied in FOP. We, the FOP Gene Therapy Working Group, the IFOPA's Research Committee, board, and staff are thrilled with the incredible progress Principal Investigator Jae-Hook Shim, who's pictured here second from the left, and Dr. Guangping Gao, pictured third from the left, and the rest of the team at the University of Massachusetts made in year 1. We hope that the FOP community is too. You will get to hear directly from Drs. Gao, Shim, and Shi, the scientist who is working on the viral vector for FOP. He's pictured second from the right. He's the delivery vehicle guy at the In Pursuit of a Cure live event on August 25th and 26th. It's exciting and daunting to see the list of all that the team at UMass plans to accomplish in year 2, the list you just outlined. But this list underscores why we have a larger goal for year 2 of the In Pursuit of a Cure campaign. By raising this money, we can go after solutions for big questions.
And back to Batman. So you're right, Michelle. The University of Massachusetts researchers have accomplished a lot in a relatively short period of time, and they do have ambitious goals. But we're still at the early stages of researching gene therapy for FOP. However, we are using today's gene therapy knowledge and the latest scientific techniques in order to pursue that. I did mention earlier that I would tell you more about Batman. It turns out that when Batman was created in 1939, he actually had a modified gene that lay dormant until 2015, until a virus-activated promoter, and we just talked about promoters, of his metagene turned on, and that's how he ended up with additional powers. So, you know, it all comes back to Batman.
I'm glad you answered the Batman question. Thank you so much for this detailed update on what happened in year 1 and a preview of what donors can help UMass accomplish in year 2. We are now going to open it up to questions. And so please ask your questions using the Q&A button. If you haven't done that, you can start doing that now. Questions from the group?
Oh, I do see some Q&A.
Okay.
Okay.
So the big question that's being answered, or that's being asked, first of all, is, if everything going on as planned, how long will it approximately take for us to get this gene therapy? When will we have some cure through gene therapy for FOP? So do you want to talk a little bit more about the process, Danielle?
Yeah, on average, it takes $1.3 billion in 15 years to go from concept to reality. That timeline is shortening quicker and quicker. And also, when we think about it, Paul Berg took from 1972, when he could inject the cells, to 1990, when Ashanti had the first gene therapy. So there is a timeline, and the timeline is mostly driven by safety, 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 safety. It's mice are not humans. We're much more complicated. So I think that it's safe to say that it's going to take a while. It's also safe to say we're moving very quickly. And I know that that's not the answer that you want, but it's just so hard to put a number on it.
Yeah. Someone asked a question about gene replacement. So someone asked, did I understand right that it's really a combination? Yeah, I don't think we don't see a name being used in the literature that it's gene combo therapy, but that's really what it is. You're doing addition and silencing at the same time.
And one of the ways you could think about it is you're replacing the activity by turning up the good and turning down the bad. What you're really replacing is the activity that should have been there all along. And one other thing to see, I see one of the questions is a cure. And we really didn't mention this, but gene therapy right now is, a lot of the questions that we have as scientists are about control. What we can't do is change every cell in your body, sort of like a scan that goes from your head to your toes that we may have seen on old Star Trek episodes. What we can do is, when we target an organ system or an area of the body, it's a little about like spraying a crowd with water. Some get a lot of water, some get none, some get some. We don't have the precise control just yet to go into your skeleton and all the cells around your skeleton and your muscles and say, let's target every single cell individually, and everybody gets one, and not too much and not too little. So what we're really going for in the gene therapy trials that are happening right now is, if you improve the genetics and therefore the blueprint in a portion of the organ, let's say it's kidney disease, will the organ as a group work better? And the answers to those questions have been, thankfully, yes. Sometimes you don't need to do curative gene therapy on every single cell. Sometimes some cells are enough.
So another question that has been asked is about sharing this video. We will make this available, the recording of this video available on our website. We'll send it out to everyone that was registered as well. So we'll do that at the end. So if you do want to share this with others, you can do that. So someone asked a question, what happens to the heterotopic bones that are already formed? Let's say, assuming that a few years from now, there is a gene therapy treatment.
Mm-hmm. Yeah, so that is a good question, because those cells have already, their fate has been determined. The Lego has been built and placed in the place it's going to, whether it's the correct place or not. That part of the story is done. However, what we envision in a big, broad sense is that at some point we will be able to do surgery to remove the heterotopic bone after someone has gotten gene therapy, so that new bone will not form.
So someone else asked the question, so if some cells are enough, then are scientists going to be focusing on the main issue, skeletal organs, to stop the extra formation of bones?
That's a really good question. There is Some people say that FOP is where your muscles turn to bone. And the answer to that is yes and no. Your muscles, soft tissues, ligaments are replaced over your lifetime with stem cells. And so what we are doing with the gene therapy is the gene therapy is on the stem cells, so that any cells that are recruited to a site of injury get the right messages and they don't all become bone.
Other questions? Someone asked you to repeat that answer.
Okay.
You could repeat that question about what happens to the bone that's already been formed.
The bone that is already formed, because the gene therapy is targeting stem cells, new cells that come on board, the bone that's already formed is most likely not going to be targeted by the gene therapy. And so what we would do is what scientists envision is being able to surgically remove the bone, the extra bone that's formed. But during your healing process, if you're given gene therapy, then your new cells recruited are not going to be driven toward a bone making process. They're going to be healthy. They're going to get the right signals. So you're going to get healing after that surgery that does not involve making more bone.
So someone asked, once the HO bone is removed, will there be muscle tissue under it, or will it have to regrow? And I think what you're saying, Danielle, is that yes, that would have to regrow, but it's going to regrow in the proper manner this time.
Yes, and what some researchers have been talking about is, we know that we don't have the surgical option right now, but let's get prepared for it. Let's remove bone. Let's use some of the drugs that are being used in clinical trials now. Let's get ahead of it so that we know what's happening to your muscles potentially before these drugs are approved, and we can move as fast as possible.
Any other questions? I'm just trying to go through the chat. There was a question about, is there already a time frame set for this research? How long would it take to start clinical trials in humans affected with FOP? Do you want to talk a little bit more about the phases? I mean, this next phase, phase 2, we'll be continuing to do this work, a little bit more about the phases that happen before we get to human clinical trials. Is there more you can say about that, Danielle?
So where we are right now is something called preclinical proof of concept. It means that preclinical is the definition for everything that happens before you get to a trial. And proof of concept is what we answered in year 1. Can you do it? And now we are in a phase, this new line of funding will come in and ask, okay, can we do it in humans? Can we do it in, do we have the best viral vector? Is it going to go to the heart, or is it going to target those stem cells that we want to target? It's very easy to target them when they're the only cells that are sitting in a dish. But can we target them in a live animal? And when we target them, do we ultimately, too many cells go to the heart, too many cells go to the brain, viral particles, not cells, sorry. So it's all about fine tuning what is safest. And that moves into a stage called IND enabling. So we go from proof of concept to IND enabling, and IND enabling stands for investigational new drug enabling. So now we're moving on a pathway to enable the technology we're using to get to a trial. And the FDA and the EMA and other regulatory agencies have very specific requirements during that phase to prove that something is safe and efficacious in humans. That's where we're going to go next. It can take, in that space, anywhere to 5 to 10 years. That's why it's really difficult to sort of put a number on it, because sometimes things take much longer than you think, and sometimes they go quicker, and we're going to go as fast as we can. As long as it's safe and effective, we're going to go as fast as we can. Once we move out of the IND enabling studies, and those are all in laboratories and in animals, then we move into clinical trials. The regulatory agencies say, yes, that you have proved enough safety and efficacy in the available models that exist, that we're going to let you go into a phase 1 safety trial. And what that is that you take the gene therapy and you put it in healthy humans, humans without FOP, and you see if they're okay. These are very low doses, and you want to know what kind of reaction. Are they going to have an immune reaction? You know, are they going to have problems with their heart? And then it goes to a phase 2. Phase 2 is this combination of, is it safe in patients with FOP, and does it work? These two are low doses. We don't want a really high dose in case it's not safe. So phase 2 is a mix of safety and efficacy. Phase 3 is where the rubber hits the road. We're going to use the dose that we think that ultimately the drug will be approved to be used at. We're going to use it in the way that we anticipate an approved drug to be used. And we're going to gather as many interested FOP patients as possible to participate in the trial. And it is at the end of the phase 3, something goes in front of the FDA or EMA for approval.
It is definitely a long process, but what I think, we want everyone to hear is that, there are steps where it's very much a stepped process. We have this idea, does it work in concept? Okay, now let's try it on FOP cells in a dish. Okay, that worked. Now let's try it on animals. Okay, now let's try and fine tune it. Let's try a little bit something different. But I think the thing that's most important to say is, this is pretty amazing biotechnology, and that safety is top of mind with every regulatory agency, whether it's the FDA in the US or the European Medicines Agency in Europe, or each country has their own regulatory body. They're going to be, monitoring the safety of this for sure.
Mm-hmm.
One other question someone asked is, can gene therapy research be pursued sequentially for silencing the defective gene and then replacing? That's pretty much what happened in phase 1. Can you talk a little bit about that, Danielle?
Yeah, so it's not so much that they're doing it sequentially, they're actually doing it at the same time. So they looked at gene addition in one half of the laboratory, and then the other half of the laboratory was looking at gene silencing, and the third part of the laboratory was looking at both of them together. And that's how they were able to work so fast within 1 year. They knew which questions they wanted to answer, and unless a specific question in one experiment was needed for the next, they did them all at the same time.
Yeah. Someone asked about using a virus as a vector for gene therapy, and I think the good news is that that's what part of the funding from year 1 paid for, was finding the right vector, the right delivery vehicle. Do you want to talk any more about that, Danielle?
Yeah, I think 2 things are really important. One, that we are using the same viral vectors of the approved therapies out there. So they have paved the road to show us that using these viruses in humans is safe. It's actually the adeno-associated virus, which we don't get sick from. It's a virus that's out there, we don't get sick from it. And so researchers really kind of grabbed onto that one and say, instead of having to fight, a bad virus, let's go get a virus that we can get infected by and we don't get sick. So they're using that one, but FOP, the ACVR1 gene, every single gene is different. And this has never been done in FOP before. It's never been done with the ACVR1 gene. So we need to know if we put the ACVR1 gene cargo or the silencing cargo or both of them into this vector, whether or not it travels through the body and gets to the right cells and doesn't get too much in one cell population and too much in another, like, if it all sort of tracked to the heart. And that's a lot of year 2. Year 2 is saying, okay, have we modified the viral vector enough? Have we modified the AAV vector enough to optimize where it goes and what it does?
So another kind of comment / question that was asked is, it looks like gene therapy would be worth it if only to stop future flare-ups, then if we can flare-ups or growth of bone, then if we can arrange surgery to correct deformities, we're good to go, correct?
Yeah, I think that that's one of the ways that researchers are looking at this. But if we're looking years down the road, and I know if you have FOP, you're not thinking about the next generation, you're thinking about, where you are today, is that when this works, so I'm going to be very careful in saying when this works, because I'm excited about it. It's going to be earlier and earlier in one's lifetime, so we won't have the buildup of deformities and loss of function. So that's one way of looking at it. As far as future flare-ups, I think that it's important to know that while folks are looking at gene therapy, there are also other drugs under investigation. I think we counted 22 the other day that are, targeted to try and stop these flare-ups. So this is we're not looking at gene therapy as the one answer or anything as the one answer. We think that we really want to bring FOP physicians an arsenal to work with and to tailor for you.
There was a question about, it says, for now it's mice, but will the patients need prophylactic steroids or immune suppression to reduce the chance of an immune response?
I think that's a possibility. That is what's happening now. But interestingly enough, we are finding that the prophylactic steroids and immunosuppressants that were used, at first with gene therapy, you don't need to stay on those forever. It's almost as if your body eventually accepts it. So in Ashanti's case that you saw, she is no longer on immunosuppressants. I don't know how long she was on them or has been off, but I know that she's no longer on them. I think we're still learning a lot to figure out what to use when.
Any other questions?
What was done with the second of the 3 mice? No, nothing. The second mouse, the one that was treated with the combination, was treated when it was very young. And when they sacrificed the mouse, they did see heterotopic bone. They did see extra bone, but they did not see bone to the degree of an untreated mouse, or bone to the degree of just addition or just silencing. So it was a really exciting, positive result, but it wasn't perfection.
Any other last questions? Someone asked, besides gene therapy, are there other medicines or medical improvements being supported by the IFOPA that we could get excited about? And I would just say, as an answer to that, that we have an incredible research portfolio of programs, and it's really your support, whether it's your donation or your fundraising activity, that's allowed us to build those things. In 2000, I've been here at the IFOPA since 2016, and in 2015, we launched the FOP registry. It is probably the most valuable research asset that the IFOPA owns. It was made possible in the beginning by support of the Radiant Hope Foundation and donations from the community. And then now it's being supported by pharmaceutical company sponsorship. But that registry is the only scientific registry of FOP patients in the world, which means that all of it is collected in one place so it can do the most good for research. So we've got the registry. We've also launched an FOP mouse model. We've also launched the FOP biobank, which makes tissues, and it makes blood and urine and baby teeth and saliva. The cells found in those things are available to other researchers to use free of charge. We now launched last year a preclinical drug testing program where if a researcher says, I have an idea, would this drug maybe work? We are able to work with the Mayo Clinic to test those drugs in mice. So there's a lot of research tools that have been developed by the IFOPA that your support makes possible. The other programs outside of gene therapy, and I mentioned one of them early on, is the Act for FOP grant program. That started in 2015, and it now funds somewhere between, we'll add some new grants this year. The In Pursuit of a Cure campaign will allow us to add some new grants this year, but we are serving more than 15 different grants through that program. One of them was launched last year. It's a pilot study, a small study that's being conducted in Israel. It's not just US-based grants, but it's a pilot study that's happening in Israel with FOP patients. So we are hopeful that later this year, we'll be able to provide you a webinar that has more information about the Act for FOP drug development program, as well as the $1 Million Bike Ride Grant Program. We, every summer in June, there's a bike race that we raise funds for, and those funds fund new FOP grants, primarily grants focused on immunology research, as well as muscle and regeneration research through the $1 Million Bike Ride Grant Program. So there is a lot of other exciting research. Gene therapy, I think, knocking everyone's socks off from what we are seeing in here. But there's also a lot of other exciting research tools that are being developed, as well as other grant programs that are making a huge difference in drug development and what we are learning about gene therapy, or about FOP. So I just want to say that you will get an opportunity if you come back and join us on August 25th or 26th at the In Pursuit of a Cure live event. You will have an opportunity to meet some of the researchers, a handful of the researchers in the Act for FOP grant program, and learn a little bit about the scientific research that they're working on. So thank you again, Danielle. I think we're at the end of our time. We are super thrilled to have your scientific expertise. I think as a staff team, we've learned more about FOP since you joined the team, because you really do a great job of breaking it down for us and helping us understand how FOP works and how these research interventions, these scientific interventions, could help our families living with FOP. So we're super glad to have your scientific experience and your experience in rare genetic diseases on our staff team here. We are also very thankful to the FOP Gene Therapy Working Group, as well as the researchers who are on the ground at the University of Massachusetts.
Thank you, Michelle. It's absolutely my pleasure. So it's an exciting time. I think we should be hopeful.
Yeah. So this is not the last time you'll get to hear from Danielle, as I said just a moment ago, in hearing about the IFOPA's research work. We look forward to having future webinars with you to learn more about FOP research and drug development activity. When you leave the webinar today, you'll be redirected to a quick survey. Please take a few moments to share your experience on today's gene therapy webinar and let us know what other research topics you'd like to learn about. We hope you'll register to join us on August 25th or 26th at the In Pursuit of a Cure live event. The event is offered 5 times over 2 days, so you can pick the viewing time that works best for you and your family. You will get a chance to meet the researchers at UMass, including Dr. Guangping Gao and the principal investigator, Dr. Jay Shim. You'll also meet researchers from around the world who've been funded by you in the IFOPA's Act for FOP Drug Development Grant Program. And finally, you'll hear inspiring stories from FOP families in our community. We want to invite everyone to hear that the FOP community is investing in progress and believing in hope. So we need you to invite your family, your friends, coworkers, neighbors, social media followers to join us for the live event on August 25th and 26th. It's easy to invite them by sharing our campaign website, which you see here, ifopa.org/curefop. We're here to assist you in sending out postcard or electronic invitations, or setting up your own personal fundraising page, like Emma did here, to help raise funds for this important campaign and fund this critical gene therapy research. If you'd like to get started, send an email to together@ifopa.org and we'll help you get started. You, the FOP community, are at the heart of all we do, and we will leave no stone unturned in our search for treatments and a cure for this devastating disease. Thank you for joining us today, and we'll see you on August 25th or 26th.